We're moving? Good. Howdy. So I forgot something. Emily's going to get it. So if you're watching this on videotape, there's nobody to operate the camera. I'll try to stay within range. I make no promises. But Emily has to go get my DNA. Did everybody enjoy yesterday? So I was babysitting the Incamas. They burnt my butt out. I was babysitting since last Thursday. My wife and I, like I was doing a lot, right? I did the wrestling on the floor part. The wrestling on the floor part. What's right, wrestling or wrestling? Wrestling, that's what I thought. Okay. How can it be incorrect? I never make a mistake. Evidently I did. Hey, how come that's not on? Oh, really? How come that's not on? One large display. Maybe I have to log on here for it to listen to me. Hey, that's not cool. That's not cool at all. So who's here for the first time? I know we have at least one. Okay? So you need to get out one of each of these handouts before you leave. Included in there is a handout to sign a form, because I don't check IDs for the exam, I check signatures. If you haven't signed your form, you don't put it off too long. Okay, so we have got an issue. Y'all are real quiet today. Y'all hung over from the break or what? Oh, that's right. You guys are Aggies. You don't do that.
West Chemist Media Center.
Yeah, I'm in Kleberg 115, and I'm dead in the water where the projector won't come on and the screen won't go up.
Okay, so you're trying to start class, though, right?
Yes.
Okay.
You're wondering if I'm trying to start class.
Okay, so I'll see if we can start it remotely, but we'll also send someone right over.
Excellent.
Yes, sir.
Thank you. Did you hear what I just said to class?
What's that?
Did you hear what I just said to class?
What'd you say?
Oh, never mind. If you didn't hear it, that's good.
I didn't hear it. Okay. Well, we will see you soon.
Okay, bye.
Bye.
I guess you need it for class, right? Oh, no. None. Nothing like that. I can't turn my phone off now. I think I turned it off. Zip, zip, zip. And I had a good picture to show you guys. One of the people that helps me in my office went to Ecuador. Brought me a rock from the equator. I'm talking the actual flipping equator. She's standing on a line that said equator. She picked up a rock from the equator. I have a rock from the equator. To me, that's just like a moon rock. I've never been to the equator. Any of you ever been to the equator? Do you have a rock? Huh? Oh, well now I've flown over it. But has anybody stood on the equator? Did you? In the middle of the ocean? How did you know you was on the equator?
Because I was on a naval ship. We parked the ship in the middle of the ocean.
There you go. Did you scoop up some water?
In my swimsuit.
Well, you should have wrung it out and kept it. So then she showed me what she ate in Ecuador on the equator. Fried? Guinea pig. I have a rule. I'm from South Louisiana, so I eat a lot of things. But I don't eat any mammal that doesn't have hair on its tail. And she said, well, a guinea pig doesn't have a tail. There you go. If it has a hairy tail, I'll eat it. No hair on the tail or no tail? So that rules out what? Possum? Rats? Except tree rats. What would be a tree rat? A squirrel, they're good. Nutra? Armadillos? Flipping guinea pigs. Oh, hey, we got it. We got it. Cool. We don't have one PC, but we got it. Let's see if I can go to one projector. One projector. Well, we can handle two projectors. We can handle this. You might go batty because you'll see two screens. Which one do I follow? Which one do I follow? Which one do I follow? They'll stay the same, so don't let your mind go crazy. It's like when you're talking to somebody. You got to focus on one eye, right? Because if you do flipping back and forth, they think you're psycho. Focus on just one eye. Focus on one side. I grew up with a fella, good friend, had one green eye, one blue eye. You couldn't focus on one eye. Wayne! It was hard. Okay, while I'm finding my bearings, who's the turkey that Snapchatted me on the bamboo stick? Snapchat's going to get me fired before this semester's over with. I promise you. So go to page 8. Emily's going to get our DNA. So we're going to skip ahead to where we're starting with whole plants. And first thing I want you to understand is how plants grow. How do we grow? Over the course of our lifetime, from being a baby to being a full-grown adult, how do we grow? In every direction at the same time. You know, that proportion stays relatively constant. Who was it that did that? Drew that? Said the width to your height to the size of everything. And that's because every part of our body grows in every direction at about the same pace, except our heads. We're born with extra large heads, and it will literally grow slower than the rest of your body. That's why babies look like they have big heads. You know why they look like they have big heads? They have big heads. You know? Plants don't grow that way. Plants grow from discrete areas at the tip of every stem and the tip of every root. Now that's their first growth. What's our term for first? Primary. So that's called primary growth. So every plant in the world has a first growth called primary growth. And if you look at the bottom of the page, a picture of a plant. By the way, I drew every diagram in this book. They might not be great, but they're the best I can do. Okay, so at the tip of every stem is going to be a bud. Okay, I got to kill this. If I open the PDF from the web page, the smart board is not smart enough to know that, so it won't work. If I open the PDF from the desktop, no, very often it will work. Let's see if it's going to be that way today. Zip, 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 and zip, and zip, and at the tip, oh yeah, of every stem is a bud. Bunch of little scales. They're really little bitty baby leaves. The tip of every stem, a bud. You may have seen a rose in bud. Okay? Every stem, inside that bud, at the tip of the stem, is an actual meristem. If I were a stem and my head were the meristem, my head would be covered by leaves. They turn brown, so they're usually brown and hard. The actual dome at the top of my head would be meristematic. It started working, but we only have one. I have it on one screen, but I'm looking at two screens. No biggie, I can live with that, but I'd prefer one screen. Oh, y'all have vests now. Yeah. Oh, that's so y'all can be recognized. Tell them you need a vest that fits and looks cool. So if I were a plant, I would only grow from the dome of my head if I were a stem. So what would I look like next year? I'd have a taller head. Everything else would be exactly the same. What would I look like the next year? I'd have a taller head. I would grow and make a big cone. How many of you know cone heads from Saturday Night Live? That would be how we would grow if we grew like plants. So you take a tree that big around. When it's young, you nail a barbed wire fence to the side of the tree. You come back 20 years later and the tree is 50 feet tall. How far apart is the barbed wire? Exactly the same. It doesn't get stretched out as the tree grows. If that was the case, every fence in the world would eventually get big gaps and the cows would jump through it. It stays exactly the same because the tree grows from the tips. The tip of every root has a meristem. So stems get longer, roots get longer from primary growth. From a meristem at the tip called the apical meristem. It's a little dome of cells. It's really microscopic. You can't even see it. Okay? If a plant's going to become woody, it will make a meristem along the side of the stem. So let's think about an oak tree. In its first year of growth, it's a little bitty oak seedling, and you have a stem growing up from the seed because of the meristem at the very tip. And let's say that first year it gets that tall. Now, it's an oak tree, so the next year it's going to want to start making a ring of wood. Actually, before the end of the first year, it'll start making a ring of wood. What does the ring of wood look like? Bark would be part of it, and inside the bark would be now think about a tree. I cut a tree down, bark on the outside, what's on the inside? Rings of wood. What would one ring of wood look like? If I could pull out one ring of wood, what would it look like? It looked like a cylinder. So if I'm a tree, I grow 1 year from the dome of my head. By the end of the next year, I want to start making my first ring of wood. A meristem has to arise to make that ring of wood. Where do you think the meristem is going to arise? If I'm a tree, what is this? If I'm a tree, I'm a model of a tree, what is this? That's I'm a young tree. First year growth, getting taller because I'm a cone head. What is this? What is this sheet? This cylinder around my body? It's epidermis. Inside, I'm going to make a ring of wood. We need a meristem to rise inside that looks like a cylinder. If it's going to make wooden cylinders, the meristem has to be a cylinder. So if I'm a young tree, what will happen is inside of me, a cambium will arise. A cambium is the name of the meristem that makes that ring of wood. Since the ring of wood is part of the vascular system, it's called a vascular cambium. Now, I will always just call it cambium, but technically it's a vascular cambium. Oh.
We got your windscreen. The ones behind it are still on, so
Oh.
we'll have to come back later and turn them off.
I am good. Thank you, fellas. Bye, IMS. So I'm a tree. I want to start my first ring towards the end of the year. I need to make a cylinder vascular cambium inside of myself to make a cylinder of wood inside of myself. What would the vascular cambium look like once it formed?
Cylinder?
Huh?
Another cylinder?
Another cylinder, and I'm wearing it. Where is that other cylinder? It's my t-shirt. Literally, a cambium will arise from the parenchyma cells inside the cortex. Some of those cells will change themselves into a meristem. So now my t-shirt is a meristem. What would happen if my t-shirt replicated itself and now I have two t-shirts? I get 1 t-shirt fatter. Then it divides again, 3 t-shirts. Divides again, 4 t-shirts. Every time it divides, one of the shirts becomes woody. The cells fill up with lignin and it becomes woody. It's a ring of cells that's now woody. One stays the cambium. It divides again, 4 t-shirts. What am I going to look like after 1,000 divisions? I'm going to be very, very fat. I'm going to look like the Pillsbury Doughboy. Now, this epidermis, it can stretch a little bit, but it can't stretch very much. You're going to reach a point where my t-shirt starts doubling, and if this shirt can't get any bigger, you're going to make me look like I'm wearing a shirt that's too little for me. And it's going to rip open. And every time it rips open, it exposes the inside. Plant can't have that. Remember, if you break the dermal tissue system, bad stuff gets in. As soon as that crack appears, it makes another meristem called the phellogen, like a size of a saucer, to plug up that hole. And that meristem that plugged up the hole divides and makes cork cells. So it seals up the hole. Tree still keeps growing. Rips over here, plug it up. Rips over here, plug it up. Rips over here, plug it up. Goes to winter. Has 1 ring of wood and a bark that's kind of brown, but kind of green. Looks like kind of scabby. Does that every year. 100 years later, you have an oak tree on campus. And you have this bark that's that thick. Have you ever noticed that it's ridged? You know why it's ridged? It's constantly breaking and exploding. And every time it rips, it's plugged up on the inside by new bark. So you end up with a really thick bark and a ring of wood. How many rings are made per year? 1. I cut down a tree, I count the rings, I know the age of the tree. Okay? And every year you'll get 1 more ring of wood and you will get a thicker bark and it will be more creviced. If you look at the redwood tree in the horticulture building, the bark is literally that thick. You know, that's 500 years of explosive growth. Primary growth from the tips of stems, tips of roots. Secondary growth from a vascular cambium to make wood and a cork cambium or phellogen to make bark. Technical name for bark is periderm. You know what we call a plant that has secondary growth that makes rings of wood? We call it a tree, and a tree would be a woody plant. Quite simply. If you see a tree that has wood, it's because it has the ability to make a vascular cambium to make the wood. Can every plant in the world make a woody trunk? Name me one plant that cannot make a woody trunk. Celery. Yes. Name me another one. Sunflowers actually can. Huh? Pear fruit can't be woody, but they can come from a woody tree. I'm talking about a plant that can never be woody. Grass. All grasses. Okay? So, let's look at the next page. And we have these various plants classified into 3 different types based on their growth habit and based on what they look like. First of all, a plant that can be woody will always be what we call a dicot or a gymnosperm. That's the technical name for the 2 kinds of woody plants. They're mostly trees, but they can be shrubs. And there can be woody plants that just live a year or 2, like a sunflower plant. Make a little bit of wood, but not a lot. There could be a redwood tree that lives 500 years and makes 500 rings. The characteristic of a dicot is it has broad leaves. They're broader, okay, than they are long. And they have net veination. The veins are crisscrossed. Here is a dicot. Is that a broadleaf? Yes, it is. So this is a dicot. This particular plant is growing in its first year. It's totally herbaceous. So this particular plant can't be woody. That would be like your celery, your lettuce, those kinds of things. Now, here is a yaupon shrub. We'll break off one leaf. There's one leaf. I know you can't see it from the back, but it's broad. And if you looked underneath and looked at the veins, you'd see they're all crisscrossed in every direction. Now, this yaupon shrub, I cut it off. It has a woody stem. This is a dicot that has the ability to make a cambium. Some dicots will be forever herbaceous. Some dicots may be woody. The difference is this plant can make a cambium and this plant can't. Some plants are destined to be woody, some are not. So these are dicots, broad leaves, net veination. The other kind of woody plant we have would be the pines and the junipers and the firs, and their leaves grow as needles, like pine needles, or scales. So if you see a plant like this juniper, where the leaves are whoop, the leaves are whoop, see if you can get me a leaf. One leaf. Just one. Oh, that's a nice little juniper branch. Look at that branch in my hand. I want just one leaf. One leaf. One leaf. Just one leaf. You've never looked closely at a juniper, have you? Oh, let me see. Oh, it's a little green speck, but it's smaller than the head of a pin. And that's a juniper leaf. The leaf is a scale. Okay? So what you're seeing is a bunch of scale-like leaves attached to every stem. A pine needle would just have long scales. Where the pine needle, so imagine a pine needle shrinks down to just a scale. So if you have scales or needles, it's a gymnosperm. It can be herbaceous or it can be woody. Can you tell the difference between a bird and a mammal? What's the main characteristic of a mammal? Hair. What's the main characteristic of a bird? Feathers. Hey, what about a reptile? When you look at a dicot or a gymnosperm, I want you to instantly tell the difference. Dicot, gymnosperm. Dicot, gymnosperm. Pine tree. Christmas tree. Okay. Oak tree. Roses. There you go. And then we have monocots. I pulled up this nut grass. It has long, skinny leaves. Long, linear leaves. You can't see it from where you're at, but all of the veins are running the length of the leaf straight up and down, parallel to each other. Long, linear leaves, parallel veination. When you see a grass-like leaf, monocot. Corn. Monocot. Bermuda grass. Monocot. Sugar cane, if you know what it is, monocot. I will expect you to tell the difference between those three kinds of plants as quickly and easily as you do birds, reptiles, and mammals. Okay? From this big old table, I want you to know the leaf type and the growth habit. And that's it. Monocots, long linear leaves, parallel veination, doomed to forever be herbaceous because it can't make a cambium. Dicots, broadleaves, net veination can be woody because it can make a cambium if it's supposed to be woody. Gymnosperm, needle scale-like leaves can be woody because it can make a cambium if it's destined to be a woody plant. Cool? I will use the term monocot, dicot, and gymnosperm from today to the last lecture of this class. Wow. Okay, now that we got my props back, what was your homework assignment? What does DNA do? So what did you decide? What does DNA do? What does a gene do? What? It's a blueprint, but what does it do? Like my grandson drew a diagram of a car, but it wasn't a car. It was a diagram of a car. A blueprint's a blueprint of the DNA. What does a carpenter do? What do they physically do? They hold nails, they hold a hammer, they nail boards together. Hold a hammer, hold a board, hold a nail, bam, bam, bam. In that context of an action, what does DNA or a gene physically do? Huh? It affects phenotypes. That's what it affects. But if I saw it in action, what would I see it physically doing? You know, if you watched a carpenter, it wouldn't take you too long to realize, hey, I know what that worker does. They grab a board, they grab a nail, they grab a hammer, and they nail it together. And then some over there, they grab a board and grab a saw and they cut them. You know? And then some of them grab a board, grab a paintbrush, and they paint it. In that context, if we saw DNA doing everything you're alluding to, what would we physically see them doing? Making proteins? No. That's the product of what they do. It tells other stuff what to do. It tells other stuff what to do, but what does it do? That's correct.
Sends messages.
Huh?
Sends messages.
How does it send a message? It does send a message. It is the blueprint, tells other things what to do, but if you saw the DNA itself, oh, maybe you don't know what it looks like. In the cell, the DNA is part of the chromosome. And if you've ever seen a picture of a chromosome in a book, it's a little fuzzy woolly bear thing. Two of them connected to each other because you have two of every chromosome. One from mom and one from dad. And that is your chromosome, and it's made out of DNA. What is DNA? It's a long linear chain of deoxyribonucleic acids. It's a long linear, that's the definition, a long linear chain of deoxyribonucleic acids, DNA. Let's see if we can kind of unravel this chromosome. Unravel this chromosome. Hey, unravel that. Oh, wop, whoop, wop, 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 wop. Wop, 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 wop. Pass it back, pass it back, pass it back. Pass it back. Pass it back. Oh, we got it. I got it hooked up on the endoplasmic reticulum there. Pass it back. I want one strand coming from me. That's good, that's good, that's good. Good. Oh, you got the hippie gene. You got the, okay, pass that back, pass that back, pass that back. No, pass it back in another direction. Whoop, 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 whoop. You got the hairy back gene. Look how long that sucker is. In the cell, the chromosome unravels, and it literally becomes that much longer than it was in the cell. The DNA is all called up like a boil of yarn. You see the different colors? Each color is a different gene. A gene is a length of DNA that codes for the production of a protein. It's a length of DNA that codes for the production of proteins. And on every chromosome, there are literally thousands of genes. This would be just one little piece of one chromosome. And each color would represent a gene that does something. So this right here is maroon. This would be the gene that makes the protein that makes you a bleed maroon Aggie. Okay, hold that. See this one here? This tie-dyed one? That's my generation. If you had this gene right here, this tie-dyed, that's what's going to make you a hippie. Okay? Now you see this gene over here? This is the gene for excessive hair production. That's going to make you a man with hair on your back. You know, a lot of men have hair on their chest, right? A lot of men have hair on their face, right? Some men have hair all over their flipping body. It looks like a gorilla. Gross and oh! That's the gene that does it. Each gene codes for some trait. Okay? And they do it by the DNA is the instructions for how to make a protein. If you wanted to know what it does, honest to goodness, I need to get some tightness here. If you wanted to see a DNA in action, it's deoxyribonucleic acid. What's its structure? A double helix. It's like a ladder that's twisted. It has two sides connected by rungs. What are the rungs of the ladder? A, T, and GC. The nucleic acids. It separates. Oh, we might have a mutation here. I gotta be able to get it all the way across. Oh, broke. Act like it didn't break. Act like it didn't break. Okay, spread it out. Yes. The two sides separate. The nucleic acids that are bound together, A with T, G with C, they're bound electrically, magnetically, by what we call hydrogen bonds, separate. One side of the DNA is the gene, one side is a fake. The sides that's the gene has exposed nucleic acids. So act like I have rungs of the ladder sticking up, sticky ends. A, T, G, C. If this is A, what will bind to it? This is T, what will bind to it? If this is G, if this is C, so what will happen is on the sticky ends, free nucleic acids in the cytoplasm will bind to their complementary pair. And that's a ribonucleic acid. It's not deoxy. Once you add all those nucleic acids on this blank chain, you have a one-chained ribonucleic acid called RNA. RNA is a copy of the DNA. We've just finished our copy. It goes back together. So if you could see DNA in action, what would you literally see it doing? Separating. That's it. So I was trying to think of a better way for you guys to understand this. And last lecture, I hate to say this, but last lecture, I went to the restroom. I probably should turn off the TV. Put no phone down. I don't want this to be Snapchatted. I go to the restroom, and I zip up my pants. Light bulb goes off in my head. Zipper! Zipper! I go to Pruitt's Fabric. I said, do you have long zippers? He said, how long? I said, any length. 2, 3, 4 feet, I don't care. And I said, I got some 18-inch zippers. I'll take every color you got. He said, I got 3 colors. Okay. I got a 3-gene hunk of DNA right here. Now, if you have long zippers at home, bring them and donate them to me. We will sew them on here and make this chain of zippers as long as that yawn. If you want to know what DNA does, when it's time to be duplicated, to make its protein, it unzips that easily. And a new zipper will bind in that spot, A with T, T with A, G with C, C with G, duplicate the mirror image, and when that breaks off, you have a one-sided zipper. That's RNA. And as soon as it is copied, zips back up. They literally go back with their hydrogen bonding. DNA doesn't do jack. It sits there in the cell in the nucleus in a very, very well protected place, waiting for it to be read. That's all it does. It unzips and zips back up. Now in the cell, it's like a big old ball of yarn. Only the part of the DNA that's going to be copied will straighten itself out and unfold. We figure, or they tell me, about 30 to 40,000 genes in a living cell, plant or animal. Okay? We have 23 chromosomes. How many genes are on our chromosomes? 23 chromosomes, 30 to 40,000 genes. How many genes on each chromosome? How many times will 23 divide into 40,000? Somebody do the math and tell me the answer. How many times will 23 divide into 46,000? Make it easy. Huh? 2,000? Several thousand genes on every chromosome, containing the blueprint, the sequence of nucleic acids, to make a unique RNA that will make a unique protein. How is the protein made? Seriously, if you have some zippers at home, donate them to HORT 201. And yeah, I can use smart, short zippers too, because genes are every length. Long ones, short ones, but I need just different colors. Anybody come from a family that has a fabric store? Anybody come from a zipper manufacturing? You know, like did your great-grandfather found these zipper manufacturing? I need zippers. Steal some zippers from your grandma. I'll pay her back. Okay. So, with that introduction, let's go back to the page where we actually have a model of what DNA does and does not do. So go to this page. What page is it? What, 7? If you don't mind, the way my brain works, the way I understand it is the bottom diagram, not the top diagram. That DNA is a blueprint. So I pictured in my mind as a rolled up scroll with instructions of how to make robots. And each robot can do a specific task. So if I want to make a robot that can saw boards, I'm going to unravel the part of the DNA that has the blueprint for making a robot with a saw in its hand. And the blueprint, the copy, is going to be slightly different because it's single-stranded, and that's my RNA. So the DNA is copied to produce RNA. Literally, that RNA comes into the cytoplasm and sits on top of a ribosome. The ribosome is the work table. And it's on the surface of the ribosome that the robot is constructed, piece by piece. So in the cytoplasm are hammers and heads and saws and trunks and tools and legs and heads with straight up and down hair. I don't know. And this diagram says, okay, go out and give me some legs and a body trunk and an arm with a saw in it and a head with long hair, and piece them together. And then as soon as you piece them together, cut them loose, send them out into this pool of robot workers. The way DNA makes is the enzyme is the little catalyst that does the work. It's a little robot that has one job and one job only. So if I were going to build a house, how many individual tasks would there be? I'm going to have one carpenter that cuts 2x4s, one cuts 2x6s, one cuts 2x8s, one cuts 1x4s, one nails small nails, one nails big nails, one does screws, one paints, one lays brick, one puts mortar. If we put the building of a house into sequential individual steps conducted by an individual robot, how many robots would it need? A lot. You'd need 30 or 40,000. And that's what it is. In the cell, DNA is instructions to make the robot needed at that time to catalyze a specific biochemical reaction. Okay? DNA doesn't do jack. DNA is a lazy son of a gun. Just sits there all curled up, all fuzzy balled. The only other thing it does is during cell division it duplicates itself. But the secret of life is in its nucleic acids. That's the genetic code. The sequence of nucleic acids is the genetic code. No two of us are alike because no two of us have the same 40,000 DNA. We have half from mother and half from father. And we're not clones. We're a hybrid of our two parents. And if you have 40,000 combinations, if two parents had 40,000 kids, you'd have 40,000 unique kids. They're all true hybrids. And we know that for a fact in the plant world. We have some plants where one plant in one flower can make 1 million seeds. And if you planted them, every plant is distinctively different in some way. Okay? What does DNA do?
Nothing. Sits there. Unzips.
I probably said that and said it wrong. DNA doesn't do much. What does it do? It unzips. My job is to give you a graphic image to not forget. Where's my oh, there they are. I get wrapped up in my DNA. So, looking at the top, what really happens is, this is what they call transcription and translation. You don't need to know that. But the length of DNA, that's the gene, uncoils, the RNA is duplicated. The RNA carries the message to the cytoplasm, so it's called mRNA, messenger RNA. The messenger RNA sits on a ribosome to expose its one-chain nucleic acids. And in the cytoplasm is another kind of DNA, another kind of nucleic acid, another kind of RNA that has 3 amino acids exposed, and at the other end it has an amino acid. 3 nucleic acids exposed, at the other end an amino acid. They will line up here in their complementary pairs. It's a tricky way of lining up amino acids in the proper sequence. When the protein is released, it takes on a 3-dimensional shape and acts as a catalyst of biochemical reactions. How can a protein do that? This is a protein. It's a long chain of amino acids exactly correlated to a long chain of RNA, which is exactly correlated with the code in a long chain of DNA. So it's a long chain of amino acids. Has all kind of funny shapes. Sometimes the chain makes a spiral. Sometimes it doesn't. But it has a unique shape. Okay? Now this spiral have a whole bunch of amino acids real close together. If they have predominantly negative charges, it'll be like the negative side of a magnet. And if you have predominantly positive charges, this might be like the positive side charge of a magnet. But your protein will never let them come together because this is its 3-dimensional shape. We may have ears that have a positive charge. if we did, this protein could pick up an ear. Now we'd have an ear sitting right there. Okay? It might very well walk up to one of those robots we were talking about who is missing an ear. I'm not going to cut off your ear, but let's act like his ear is missing. It could walk up, diffuse up, get in the right position, and if you had a greater negative charge than the protein, you can suck that flipping ear off. Now you got an ear. And that's how proteins catalyze reactions to build something. But we might have another enzyme with another shape. Are we back on? Hey, I really apologize. First lecture, I tell you, we're not professionals. Okay. So what I want you to know for the exam, basically what is on this page. The definition of DNA and RNA, the definition of nucleic acids. I think you already know that there are 4 nucleic acids in DNA, A, T, and G, and C. I want you to know the pairing. I want you to know that in the RNA, there is no T, but U takes its place. So in DNA, A binds with T, T binds with A, but in RNA, A binds with U, U binds with A. Know the pairing. Know the definition of a gene, a length of DNA that codes for the production of a protein or a part of a protein subunit. I want you to know the protein is the product of decoding DNA. Some of those proteins act as metabolic catalysts, and when they do, we start calling them enzymes. Every characteristic about you is because you had the DNA to code for the protein to make whatever it is we're talking about. We all started out as a single cell with a single nucleus with 2 of every chromosome, 1 from mom and 1 from dad. We'll see later in the semester when that happens, that fertilized egg is called a zygote. You don't need to know this right now. We all started out as a fertilized egg, because I don't think we have a clone in this room yet. We all started out as a fertilized egg. That egg divided to make 2 cells, then 4, then 8, then 16, then 32 by normal cell division. Every cell in your body is identical genetically to the original zygote. Every cell in your body has all the DNA to do everything. But it's all controlled. So it only does things when and where it is needed. You know, think of DNA expression as an orchestra. And DNA is instructions for making the orchestra members. And if you want to play a song, every person playing every instrument has to play the right note at the right time. The simplest, and I could do this, the simplest is the cymbal player. Got to be at the right place, right time. No, idiot! No! That's a mutation. Expressed at the wrong time. You know? And that's what mutations are. Expressing the wrong gene or just the same gene at the wrong time. Every cell in my body has instructions to make a nose. Somewhere on the beginning of this little primordial face, those cells said, let's make a nose. And they made the proteins that make nose. What freaks me out is that these cells can do the same thing. Oh gosh, I hope they never do it. This DNA stuff is so tightly controlled, that's the marvel of a living organism. Okay? But you are a product of your DNA, your genes, and the proteins that are expressed. In your lifetime, scientists will be able to very easily clone DNA, move it around in new organisms, and do genetic manipulation. The first expression will be in medicine. Gene therapy. The ultimate of that is cloning a human being to make a duplicate. That's the ultimate expression. You will see all of that continuum. You have to decide what's right and what's wrong based on your morals, your ethics, your religious beliefs, the way you were brought up, et cetera, et cetera, et cetera. What is 2 for? We already have a person in China who supposedly has cloned a human being. Not completely, partly. We actually had somebody who did it earlier. They cloned everything except the mitochondrial DNA. So we already have scientists that can and maybe, or trying and maybe have done that. You have to decide if that's right or wrong.
Don't they like grow ears and stuff like that? Like
They're trying to grow, they're trying to manipulate genes to grow ears. Yes. You know? Maybe they can take some fat cells out of me. I got plenty of that. Change the DNA so now those cells can make insulin. Put them back in. And now cure diabetes. Give them to you? No, you have no diabetes. Would you have a problem with that? Maybe not. But cloning yourself. Would you have a problem with that? I don't want to be cloned. My philosophy is very simple. We need only one Dave on this planet. There are people on this planet that are having their whole bodies frozen with the idea that one day when we can clone, they will thaw them out and clone them. And if you don't have, this is true, if you don't have enough money, they will freeze only your head. Gross. Exactly. I can't picture myself in liquid nitrogen. Hey, no, we don't need to go there. Okay. Okay, so we did that. We did that. Took me a little longer than expected to do all that. Y'all had so many questions. It's up to you. It's up to you. Close? That's a negative. I don't respond to negativism. I don't respond to negative. I respond to being buttered up. Yes, I do. We're going to omit that. We're going to omit just some of it. But some of it I want you to know. So now we're starting our anatomy lectures, where we look at the outside of something and we name what we see. And then we're going to cut it open and look at the inside of something and name the different parts that we see. We can begin looking at morphology and anatomy. So this is a stem. Okay? Some of this you already know. That at the end of every stem is a growing point, and we call it a bud. Since at the tip, we call it a terminal bud. It's a meristem surrounded by those little scales. Also, stems have leaves. So if I want to draw the leaf, it's going to be attached right there. And the leaf and the buds are attached at the same spot, and we call those nodes. So a stem has joints like the knuckles of my finger, and that's a node. Everything is attached to the node. The leaves are attached to the node. On the side, there's a bud attached at every node. In the angle of every leaf, we call it the axil. The axil of every leaf is a bud. So we have terminal buds and we have lateral buds. The region between nodes, an internode. So know that stems are made out of nodes and internodes, everything attached at the node. Know that there's a bud at every node. What happens if I cut off the top of that stem and throw it away? What happens if you prune the tip of a stem? It starts branching. The lateral buds will start growing. When we talk about pruning at the end of the semester, that's the basis of pruning. When we remove the terminal bud, those lateral buds will start growing. And it could be a 100-year-old oak tree. Cut an oak tree down, you'll see the sprouts coming from the base. Those lateral buds are to rejuvenate and replace the plant when the top is broken down. Okay? So know that parts of the stem. The buds at the tip in every node. At every node is a leaf and a bud. And the lateral buds grow when we prune the tip of the plant. Cool? So what I want to do is take the different kinds of plants we talked about, take the stems, cut them, and look at their cross section. Now, I will tell you right now that dicots and gymnosperms have the same kinds of stems. Internally, even though the outside looks very different, internally, they have the exact same internal anatomy. So let's take this as a young dicot stem. Let's take this as a young dicot stem. Let's pop off some leaves to give me some room to work. Okay, let's take that stem and let's make a cross section. Look at the cutting up. Okay, if I cut my finger and you looked at the cross section, what would be in the very center? A bone. What would be around the bone? Muscles. What would be around the muscle? Skin. Tissue called skin. You think you might see the cross section of some veins? And arteries? Spitting out blood like crazy? Yeah. So let's cut this stem and look for this. So remember, my job is to give you an image that you will not soon forget. So let's cut a stem and let's look at the cross section of that stem. Now I'll tell you right now, on every plant part that I talk about in primary growth, every plant part in primary growth, whether it be a stem, a leaf, a root, a flower, a fruit, a seed, every plant part where I say it's primary growth, the outer ring of cells is going to be an epidermis. That's the skin of a young herbaceous dicot or gymnosperm. And then inside, you will see vascular bundles, a bundle of vascular tissue arranged in a ring. And inside each vascular bundle, you'll see the two conducting tissues, xylem and phloem. Xylem will always be on the inside. Phloem will always be on the outside. Now, since as a ring, we have a part in the middle called pith. And between the ring and the epidermis, we have a part called cortex. We have two zones that aren't named, so we can call them pith and cortex. They're primarily going to be parenchyma cells. So when you eat a celery stalk, you're mainly eating the parenchyma cells between those strands of vascular bundles. When you ate that apple, there were vascular bundles in there you couldn't see. Oh, our apple person's gone. Oh, you're here. There was vascular tissue in there you couldn't see, and you're eating the parenchyma cells around the outside. Okay? What would that look like in 3 dimensions? Somebody make me a model in 3 dimensions. I want to see a 3-dimensional model of that. Very simple 3-dimensional model with what you have in your possession right now. Okay, close. Okay, take the top off. Okay, hold it up. So you're saying it's going to be a cylinder. What's the cup? It is the epidermis. Okay, take that bark off. Yeah, see, he's trying to go secondary growth on me. No, yeah. We need some vascular bundles. Give me some vascular bundles, going straight up and down in a ring-like fashion. Need a vascular bundle. Help him with some vascular bundles. Yeah, he got idea. Give me some vascular bundles. Huh? A pin. Give me. Do it. A pin. Okay, I need 6 of them. Inside. The bundle has to be inside. Somebody hold them for him. No, we need some holders. Come on. 4 people holding them. Hey, 4 people. No, 4 people. Get up there and hold them in a circle. Seriously, get your tails up there and hold them in a circle. Okay, give me some more people holding them in a circle. There you go. Okay, who has some chiclets? I just spit. Jelly beans. Who has some candy? I need some candy. I need a bag of candy. Peanuts, M&Ms. I need a bag of something. Bag of little bitty things. Come on, bag of little bitty things. Candy, chiclets, M&Ms, Eclipse mints. Can I have them? Dump them in there. Dump them in there. I need more stuff. I need more stuff. I need more stuff. What you got? Yeah. Throw him. Well. Okay. Oh, put them in there. We got more stuff? Anybody got a big bag? Okay, if we filled the cup up with little bitty things, what would that be? What are we putting in there? What kind of cells? And made out of parenchyma. Can you eat it? Can you eat it? Can you eat it? That's why I wanted candy and food. That's exactly what a primary stem would look like in 3 dimensions. The epidermis is a cylinder. The epidermis is a cylinder. The vascular bundles are like pins going straight up and down. And the filler tissue around it are your parenchyma cells. Good, thank you. That's great. Okay. Woody plant. Oh, you can put it down, but don't put it away. Woody plant. If I'm a herbaceous plant, and I'm a dicot or a gymnosperm, I'm going to have those tubes running straight up and down. Okay? Think of it as like veins coming straight up and down, arteries coming straight up and down, my xylem and phloem. They're going to be arranged in a ring. I'm covered by an epidermis. Inside, if I'm going to become woody, a cambium arises, which is now a cylinder on the inside. I need a slightly smaller cup. Put it in there. Okay, that could be my cambium. If the cambium starts dividing, it's going to make rings of wood and it's going to grow out. Okay? So if we wanted to, we could make some wooden cylinders and put them inside the cambium. And every time we did, it would have to expand out. Then another wooden cylinder, not plastic, but a wooden cylinder, put it inside. Now we're starting from a little bitty pith, surrounding a pith. Then another ring, then another ring, then another ring. Ooh, which way is the plant growing? In secondary growth, which way is the plant growing? Out. In secondary plant growth, plants still grow away from themselves, but in width. After 3 years, I'll have 3 rings of wood. The original pith in the middle, which is now going to be just a little speck. Outside of that will be a soft tissued cambium. The cambium also on the outside, every now and then, when it divides, the outside layer becomes phloem. But not a lot of phloem. Phloem never gets woody. So after 3 years, we have 3 rings of wood on the inside, be like 3 cups inside, and then a cambium on the outside, maybe like stretchy fabric, I don't know. And outside of that, maybe a soft material, that would be the phloem. And every time it expanded, the epidermis ruptured and was plugged up with bark. Oh, put the bark on there again. At least we can see the bark. You see the bark? As that whole cup got bigger and bigger, a bark formed to seal it off. So in secondary growth, I'm going to see 1 ring of xylem, wood, for every year of growth, surrounded by a cambium, surrounded by some phloem, surrounded by bark. Where's the cortex? It's crushed. Between the inside xylem growing and the bark growing, it literally gets crushed to nothing. Cool? See you next time, peep. Y'all were good today. Thank you. Oh.